All right, so this is part 3 of chapter 4. We're starting with endocytosis, moving cells across the membrane, using the membrane. Sorry about that. So we're using the membrane, we're creating membrane-bound compartments in some transport. So endocytosis, exocytosis, these are all forms of transport where we use a membrane. Why this kind of transport? It's to transport macromolecules. So the things that we've been talking about so far have been transporting very small, maybe monomers, glucose molecules, or amino acids, or ions. But now we want to transport maybe entire proteins, or we want to transport a bacterial cell, or something that's really large that will not be able to fit through a carrier protein or a channel protein. So the cell will use membrane compartments to do this. Endocytosis will go through 3 kinds of endocytosis, and then there's exocytosis. So let's start with endocytosis. So there are 3 kinds of endocytosis. Endo meaning bringing things in. The first one we're looking at is phagocytosis, which is cell eating. So the cell is going to bring in large solid particles. We can see here there is a distinct particle. The membrane is going to form an invagination to surround the particle, trapping the particle in, and then this will fuse together and bud off. So we're going to have something called a phagosome. The phagosome is basically just that vesicle which was created immediately after bringing the particle in. Then, if in order to digest, if we want to digest this particle, then we know we need a lysosome to come and fuse with our phagosome. The lysosome, remember, has enzymes inside, hydrolytic enzymes, and also a low pH, so an acidic pH. And so these are going to be degradative, right? They're going to break down and hydrolyze the macromolecules of our particle. So they're going to fuse, and the fusion will create a phagolysosome. And that's telling you that now the contents of the phagolysosome are being digested. So we have fragments created. So this is something to keep in mind, because just because the cell brings something in inside a phagosome does not mean that it's dead. It does not mean that it's digested. It has to fuse with a lysosome in order for killing to take place. So if you take microbiology, there are some bacteria that can be brought into the cell with endocytosis, but can survive the phagosome. They actually break out and they enter the cells, or they don't get digested. So anyhow, this is just one thing to think about, that we have fusion with the lysosome in order to digest those substances. Pinocytosis. This is pinocytosis. Oh no, this is exocytosis. I don't have a picture of pinocytosis. I'm sorry, guys. Well, pinocytosis is cell drinking. So we're bringing in particles in the water and continuously taking sips of the extracellular fluid. And this is also non-specific. So whatever the cell takes in, it's basically just like phagocytosis, except we're bringing in substances in the extracellular fluid. So it's called cell drinking. It's just constantly taking sips of the external fluid, the extracellular fluid, bringing it in. The one here is our receptor-mediated endocytosis. And receptor-mediated is our most specific kind of endocytosis, because there are receptors that are put in place. And these receptors are proteins that have a binding site that will only bring in a certain kind of molecule. So receptors for a certain kind of molecule. Okay. So that way the cell knows exactly what it's bringing in. What happens in this process is that there is a molecule called clathrin. Clathrin proteins will actually form the pit around the receptors. And when the receptors bind, it will allow this basically phagosome to be created, right? So we have a receptor protein first, the molecule that is going to bind to the receptor protein. We have receptor proteins in high concentrations around a clathrin-coated pit. We can see that the clathrin is causing the cell membrane to form a deeper invagination, eventually budding off into our vesicle here. So it's a membrane-bound vesicle. We do have the molecule that the receptor is bound. We can see that other molecules got trapped in there as well, but what the cell is really trying to do is bring in that purple triangle, or the molecule that bound to the receptors. Okay, so we call those a clathrin-coated pit, and we can see that the clathrin proteins get recycled. They can go back out to this plasma membrane to create another clathrin-coated pit. All right. Exocytosis is just the opposite of endocytosis. So we'll have some kind of molecule that wants to be secreted from the cell, right? So this is a molecule that's made, maybe synthesized internally, or it could be a waste product from metabolism. But whatever it is, the cell wants to move it out and push it out into the extracellular fluid. Again, these could be hormone molecules, they could be enzymes, or they could be waste products. So the secretory vesicle, right now it's called a secretory vesicle because it's going to secrete whatever the contents are, will just come up and fuse with the plasma membrane, and we can see the release of contents after it fuses. All right, moving on to our last topic here of epithelial transport. We're moving substances across an epithelial membrane, right? We want to look at just some terminology, just refresh our memory of what is an epithelial membrane. So we have the lumen, which is going to be the space that the epithelial cells will face. Remember, the epithelial tissue is a lining, right? So one side of the epithelium is going to be either outside your body, or it's going to be a cavity. It's facing some kind of lumen. And then the other side of our epithelium is going to be facing the basement membrane. So remember, that is the membrane that anchors the epithelial cells to the connective tissue below. In the connective tissue below, we have our blood vessels. We have fluids surrounding our blood vessels and surrounding the tissues. So it's called interstitial fluid. And the membrane that faces the bloodstream is going to be called the basolateral membrane. So baso is bottom, lateral are the sides. Either way, the sides or the bottom, right? If a molecule moves past either the sides or moves through the bottom, we are making our way into the interstitial fluid and then into the bloodstream. So the basolateral membrane is put together as one because that membrane, it doesn't matter if it's the sides or the bottom, it can actually reach the bloodstream. Now, remember that there are tight junctions in place to seal the epithelial cells together. So we're making a sheet of cells, a membrane, and these tight junctions make the cells impermeable to substances. So substances do not go through the tight junctions. So substances are controlled if they go through the membrane by what's on this apical surface. So the apical membrane, the membrane that faces the lumen, is going to have specialized carrier proteins and specialized channel proteins and all those active transport that are going to be specialized to move substances from the lumen into the cell. On the basolateral surface, it's going to be specialized for pumps and mechanisms that move things inside the cell, outside, so it can reach the bloodstream or the interstitial fluid. Okay, so we look at the outline, we see that only the basolateral membranes will have a sodium-potassium pump. So there's our sodium-potassium pump. And it's the same pump we talked about. Three sodiums are moved out, 2 potassiums are moved in. They also have leak channels, potassium leak channels, and we also have a glucose carrier. On the apical surface, okay, we're going to have sodium channel leak proteins and a sodium linked glucose pump. Okay, so let's talk about the apical surface. So the apical surface here is the mechanisms of epithelial solute transport, right? We can see that we have on the apical surface the sodium channels. So sodium is going to come in from the apical surface, and it's going to move out the basolateral surface. Now, what is going on here, right? This is going to be a transport mechanism that uses ATP, so it's moving sodium out of the cell and potassium into the cell. What is this called? This is our sodium-potassium pump, right? So it's primary active transport here. This is our sodium-potassium pump. So this is a way of moving salt, right? So this is if you eat potato chips. It's moving salt from the lumen into the cell, through the cell, and out the other side, and so the salt reaches our bloodstream. The potassium pump, the potassium enters the cells on the basolateral surface. We can see that there's also a leak channel protein here, a leak potassium channel. Okay. So this is your symport system, right? What's going on here is that we're using our sodium being transported in down its concentration gradient. Remember that sodium, right, is at a higher concentration outside the cell than inside the cell. So this is moving down its concentration gradient into the cell. At the same time, we're moving glucose in the same way into the cell. So what kind of transport is this? This is going to be secondary active transport. We're using the concentration gradient set up by the sodium-potassium pump, which uses ATP, but we're using that as potential energy to also help move glucose against its concentration gradient, right? So we can see that glucose is higher in concentration inside the cell. Now, the concentration gradient is higher inside the cell. We notice that we are allowing glucose to leave the cell on the basolateral side so that the glucose can reach the bloodstream. In this case, this is basically passive transport, right? Movement of glucose from inside the cell outside, that's going to go down its concentration gradient. And so we have passive transport there. All right. So in terms of sodium transport, there's a low intracellular sodium concentration, right? This is caused by an outward pumping across the basolateral membrane. It creates an inward gradient, right? So sodium wants to move in. The sodium leaks across the apical membrane. All right, so why do we have this on the apical membrane? Because we're using this to power your symport system with glucose, right? And glucose is going to be available in the lumen. So when you eat food, that's where glucose is going to be found, like the new glucose that's made in the body, that's coming into the body. Okay, so sodium is transported across the cell because we can see that sodium also exits the cell in this sodium-potassium pump. All right, so the glucose transport, this is secondary active transport on the apical membrane. We looked at this. And then we have on the basolateral membrane, we have facilitated diffusion, right? So that was how the glucose left the cell and entered the bloodstream. It was diffusing down its concentration gradient, but through a carrier protein. So glucose is also transported across the cell, right? So let's take a look at that, right? Glucose comes in the apical side, goes out the basolateral side. All right, so water transport. So solutes are pumped into the interstitial fluid. Osmotic pressure of the interstitial fluid will increase, and then water is osmotically pulled across the cell. Water is transported via the lumen into the interstitial fluid. All right, so let's take a look at water transport. So what we see here is the lumen, and we see our interstitial fluid. And we see that the solute molecules, what's happening is there's arrows moving the solute molecules into the interstitial fluid. And where the solute molecule goes, water will flow, right? So if there's a greater osmotic pressure for water to move towards the interstitial fluid, water will flow from the lumen, right, through the cells into the interstitial fluid. So it's moving towards this hypertonic environment, this environment set up by pumping out the solutes. Okay, so one side of the epithelium faces the lumen, and the other side, referred to as the facing membrane, faces the interstitial fluid. Which of the pairs below is a correct association between the two sides of the membrane and the term used to describe them? So take a look. All right, so what faces the body lumen? This is going to be the apical surface, right? So we can cross out C and we can cross out A. And then the blood facing is the basolateral surface. So your answer is D. All right, so this is just a quick look at cystic fibrosis. Epithelium does not transport enough solutes into the interstitial fluid because there's a defect in the sodium channel. Epithelium does not transport enough solutes in interstitial fluid. Active transport solutes from the lumen to interstitial fluid, creating higher concentration of solutes. Yeah, it draws it in one. Okay. So in cystic fibrosis, what we have is a dysfunctional, oh, sorry about this. Let's go back to this view here. What we have is a dysfunctional sodium transporter. So the epithelium does not transport enough solutes into the interstitial fluid because there's a defect in our sodium channel. And water does not enter the tissues. And so if water doesn't enter the tissues, and so this is a little bit flipped from what we're talking about, but water, so we can just see the general trend where sodium is being pumped out, and wherever sodium goes, water will follow. And then the chlorine here is going to follow sodium because it is a negative charge and is attracted to the positive charge as it leaves. So if there is a sodium pump that's not working, the chlorine is not exiting, we don't have a lot of solutes outside, and water is also stopped because there's no osmotic pressure to draw the water out. And so the mucus of people with cystic fibrosis gets extra thick. Right here, the mucus is diluted with water. So the mucus is watery, and it's able to move through your lungs with the cilia that lines your respiratory tract. But in cystic fibrosis, there's no water to dilute the mucus. The mucus becomes very thick and very heavy, and it's not able to be moved through the respiratory system. And so it can also coat the lungs and make it difficult to breathe by narrowing those bronchi and the bronchioles of the lungs. So breathing is hampered. And also the stasis or the static, the not moving those substances up out of the lungs can also lead to infection. So this really thick mucus is really difficult to move. Coughing doesn't move it. And sometimes they have to use just beating on the chest and some other methods to help to move the mucus out of the lungs of someone with cystic fibrosis. And our last word here is just transcytosis, which is just the process of moving a molecule across the entire cell through endocytosis at the apical end and exocytosis at the basolateral surface. So if it moves throughout the entire cell, that is called transcytosis. So endocytosis at the top, exocytosis at the bottom, that's transcytosis. And that's the end of part 3.